Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about cell division and differentiation. So with respect to the topic at hand, we can divide cells in the human body into two major categories. The first category is made of somatic cells. Somatic cells, or body cells, for the purpose of this discussion, the most important thing to understand is that these are cells that are diploid, meaning that they have two copies of every chromosome. That gives them a total of 46. Now, when somatic cells go through the cell cycle, they tend to undergo mitosis. So they undergo mitosis for the sake of growth and repair, not really reproduction in that case. So the other type of cell are what we call germ cells. Germ cells are present only in the gonads. They are also diploid. However, these germ cells will produce cells that then undergo meiosis to produce haploid gametes, gametes being sperm and egg, which have one copy of every chromosome. And this actually is for the sake of sexual reproduction, whereas with mitosis and the somatic cells, it's mostly just for growth, repair, and those sorts of things there. So the cell cycle itself is going to include all of the activities that a cell undergoes to both prepare for and execute mitosis and meiosis, whatever the goal of that particular cell may be. So the cell cycle consists of interphase, which consists of the two gap phases, G1 and G2. These both involve the synthesis of raw materials such as proteins, lipids, membranes, organelles, and things of that nature that are going to be necessary for dividing the cell. Interphase also includes an S phase in between the two gap phases. Most importantly, this involves one round of extremely accurate DNA replication. M phase, or mitosis, involves the divvying up of duplicated materials into two daughter cells, which are then separated by cytokinesis. The illustration of the cell cycle that you're seeing here is for mitosis, but for meiosis, there would actually be two nuclear divisions rather than just one. So if we look at a little flowchart here that should illustrate some of the similarities and differences between mitosis, at least how they are relevant for human physiology. For mitosis, we start with a diploid somatic cell. It undergoes S phase to temporarily become a tetraploid cell that has 4 copies of every chromosome. Then, after everything else, it then eventually goes through mitosis and cytokinesis to then produce 2 genetically identical somatic cells, which are both diploid, and these 2 somatic cells should be genetically identical in every way to the cell that begat them. Now, meiosis is a little bit different. Those germ cells, which are a kind of stem cell that we'll be talking about later, produce a type of cell called a primary gamete. In men, these are called primary spermatocytes, and in women, they are called primary oocytes. So, these primary gametes are diploid, but they're going to go through meiosis. This is going to involve one round of DNA replication to become a tetraploid cell. The first round of meiosis, which includes the first nuclear division, will divide them into secondary spermatocytes and secondary oocytes. These are both going to be diploid, but instead of just leaving it there, we are going to have a second nuclear division that is then going to produce mature gametes, which are each going to be haploid, having one copy of each chromosome. And then importantly, because of crossing over and because of independent assortment mechanisms that you probably learned about in general biology, each of these gametes is going to be genetically unique, or at least as close to unique as humanly possible. Okay, so let's talk a little bit more about cell cycle regulation. The cell cycle includes a number of guardrails to make sure that cells only go through the cell cycle when conditions absolutely call for it. To this end, these guardrails include a number of checkpoints that prevent or even inhibit progression when conditions are not favorable for a cell going through this. This includes a checkpoint at the G1 to S transition. In this case, the cell is going to check the status of the DNA to make sure that the DNA is not damaged or anything like that before we go through the process of trying to replicate it. There's a checkpoint at the G2 to M transition to make sure that everything is all good to go before we divvy up the materials and divide them into two different cells. And then in the middle of mitosis, there is what is called the spindle checkpoint. This occurs at metaphase of mitosis, and this is a check to make sure that all of the chromosomes are properly aligned in the middle of the cell. So the idea here is that if any one of these checkpoints is failed by the cell cycle will be arrested. The cell will be given a short chance to correct whatever the problems are, but if those problems cannot be corrected, the cell cycle is aborted entirely, and in most cases, the cell will be destroyed via a cell death mechanism, which we will talk about soon. So cells in the body, those somatic cells, most of them actually spend most of their time in a state of quiescence, which is sometimes called G0. This signifies a temporary exit from the cell cycle, but in many cases these cells can be induced to re-enter the cell cycle with proper stimulation, which is going to include things like growth factors, hormones, and things of that nature. A lot of other cells are actually what we call post-mitotic, meaning that they have actually permanently exited the cell cycle. Some good examples of these are brain and heart cells. So you've probably heard of the fact that you're born with a certain number of brain cells, you're not going to make any more. Same thing goes with the heart. If somebody has a stroke or a heart attack, you can kind of understand why that's such a big deal, because since those cells are post-mitotic, if you lose any of those cells, they will never, ever, ever come back. So again, that's why strokes and heart attacks, which affect the brain and the heart respectively, are such big deals, because those cells cannot be regenerated by mitosis. Okay, let's talk a little bit about cancer. So basically what you're going to find here is a lot of the regulation of the cell cycle and a lot of those cell cycle checkpoints are basically there to make sure that we don't get cancer. But obviously people do get cancer, so it's worth talking about those checkpoints and how certain things can go wrong with those checkpoints and eventually lead to big problems like what we see with cancer. So the body maintains homeostasis of the total cell number, which is for most people about 30 trillion or so, by striking a balance between new cell division and cell death, the most common type of cell death being apoptosis. So if someone is going to maintain a constant number of cells in their body, we need to have a good balance between new cells being created and old cells dying off. So this homeostasis is largely achieved because of that tight regulation of the cell cycle that we talked about before, having proper oversight of cell cycle progression with those checkpoints. But the problem is that if something happens that causes those checkpoints to fail, you're going to end up with uncontrolled cell division, meaning that cells in the body will enter the cell cycle even when they're not supposed to. So if that's the case, the total number of cells in the body is going to greatly increase because the effect and influence that mitosis has is going to greatly outweigh the influence that any normal cell death has. So to that end, there are two different types of genes and proteins that regulate cell cycle progression and help to maintain the integrity of those checkpoints. You have proto-oncogenes. These are proteins that help to promote entry into the cell cycle in response to things like growth factors. So this is going to include not only the growth factor hormones themselves, but growth factor receptors, signal transduction proteins that help to relay the growth factor signal to the inner parts of the cell, and all the proteins that play some role in getting the cell to enter into the cell cycle. A good way of thinking about proto-oncogenes is that these are proteins that have a positive influence on getting the cell to divide. And then on the other hand, you have what are called tumor suppressor genes. These are going to have a negative influence on the cell cycle. They inhibit entry into the cell cycle when conditions are not favorable or checkpoints are failed. So tumor suppressor genes are going to prevent a cell from going through the cell cycle when they are not supposed to. So the idea with cancer is that you often end up with genetic mutations that reverse the intended or normal function of these genes and of these proteins. So in the case of proto-oncogenes, their function becomes compromised because of genetic mutation, and usually this is going to cause the proto-oncogene to turn into what is called an oncogene. An oncogene is a protein that is going to push the cell into the cell cycle even when that cell should not go through the cell cycle. So this is sometimes what we call a gain-of-function mutation, a mutation that causes a protein to either become way too active or it causes a protein to have a new function that is not a good thing in this case. And then with tumor suppressor genes, you'll recall that these are the ones that prevent the cell from going through the cell cycle. When their function becomes compromised due to mutation, it's usually because they lose function. These are called loss of function mutations. Their function, because of the mutation, is totally lost, meaning that they are not able to stop the cell from going through the cell cycle in cases when they shouldn't. Now, as far as cancer goes, we're not going to turn this into a whole discussion on cancer, but usually with cancer, it requires multiple mutations to multiple genes before a cancerous neoplasm can actually form. So you would have to have probably at least a mutation to both a proto-oncogene and a tumor suppressor gene at the very least. Okay, let's polish off this discussion by talking a little bit about stem cells. So most of the cells in the body that actually regularly enter the cell cycle are called stem cells. This includes the germ cells that are present in the gonads that we talked about before. So stem cells are just non-specialized cells that can differentiate into specialized cells if conditions allow for it. So in the coming few videos, when we talk about muscle cells and nervous system cells and epithelial cells, we're going to see that all the different cells in the body look very different compared to one another, and as such, they have very, very, very different functions. So those specific types of cells are the result of differentiation. This is the process of a cell changing its shape and changing its function to fit what is intended to do with respect to physiology and homeostasis. A stem cell is a non-specialized cell that basically is kind of like putty. It's kind of like a blank slate. It's not good at doing any one particular thing just yet, but it has the potential to differentiate into something more specific like a neuron for sending electrical messages, a muscle cell for contracting and relaxing, or things of that nature. So there are 4 basic types of stem cells, and we can classify these stem cells based on how much potential they have. The stem cells that have the greatest amount of potential are called totipotent stem cells. Totipotent stem cells can differentiate into any kind of cell that you can find in the body, and they can actually support on their own the development of a new organism. The only place you're going to find a totipotent stem cell is in a zygote. So when you were a single-celled zygote, which is the product of fertilization of sperm and egg, you have not only the potential to make all the different cells in your body, but that one zygote was on its own capable of developing and growing into a single human. Pluripotent stem cells are also very high in potential. Pluripotent stem cells can differentiate into any kind of cell in the body, but they on their own are not sufficient to support the development of an organism. A good place of finding pluripotent stem cells is in an early developing embryo. Multipotent stem cells have less potential. They can differentiate into just a limited handful number of different types of cells in the body, and a good example of these are hematopoietic stem cells, which we will provide an example of here in just a moment. And then unipotent stem cells have the least potential. These are stem cells that literally can only differentiate into one very specific kind of cell. And depending on what kind of stem cell you're talking about, this is very much predetermined. You'll often find unipotent stem cells embedded in the deeper layers of your skin. This is why when you cut yourself, these stem cells activate themselves and are able to regenerate the lost skin cells and lost epithelial cells that you've lost, but that's all that they can do. They can't make other types of cells as well. So let's revisit that one specific case of multipotent stem cells. So hematopoiesis is the production of blood cells that occurs by stem cell division and differentiation. This is going to produce all of the different blood cells that you will become familiar with as the semester goes on: red blood cells, the various types of white blood cells, platelets, and so on and so forth. So these hematopoietic stem cells are embedded in red bone marrow, which is only found in long and flat bones in the body like the femur and the sternum. So if you look at this diagram, this is what is called a lineage map. Lineage maps helped let us trace the potential places that multipotent and higher potential stem cells can go with differentiating into different types of cells. And oftentimes the route that they take is going to be determined by small molecular cues like hormones and paracrine signals. So at the top of this lineage map, you see a hematopoietic stem cell, which is also called a hemocytoblast. It can go through one of two different lineages. It can go through a myeloid lineage or a lymphoid lineage. If it goes through the myeloid lineage, it can be used to produce more red blood cells, which is often very necessary because red blood cells don't live more than just a few months, so they have to be constantly regenerated so we can carry oxygen throughout our blood. Other types of myeloid cells, which we're not going to talk about just yet, they can become megakaryocytes, which are big, large cells that then fragment off to form platelets. We can form a large number of our white blood cells, all of this in the myeloid lineage. And then in the lymphoid lineage, we can form some of our more specialized white blood cells, like natural killer T cells, and then some of the antibody and antibody-recognizing lymphocytes. Okay, that's going to do it for this video. Here is a list of vocabulary terms you might want to be aware of. And then for checking your understanding, number 1, why must entry and exit from the cell cycle be under very strict regulation? Number 2, what happens when cell cycle control proteins become mutated? And number 3, how does the body replenish red and white blood cells? Okay, that's going to do it for this video. If you have any questions, go ahead and drop them in the comments section. I will see you next time.